Hi guys, am I audible? Hi people, I hope I'm perfectly audible and visible to all of you. If it is so, do let me know in the chat section, guys. Hi everyone, am I perfectly audible and visible to all of you? Yes? Am I perfectly audible and visible to all of you? Okay, that's great. Okay, that's great. So, welcome everyone. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and the chapter that we are going to start is cell cycle and cell division. Right? We have completed the chapter cell. The remaining portion of cell was completed on Unacademy platform in our special classes. I have completed that. Right. Once again, we will also discuss the cilia and flagella. That too, you guys can check on Unacademy platform, and you can just search my name. You can just subscribe to my profile, and there you will get the classes. Okay, there you will get the classes, and that class is going to be free of cost. Okay, fine students. So that is it. So today the chapter that we are going to start is cell cycle and cell division. Right, the next chapter that we are going to start is cell cycle and cell division. We'll finish the cell biology, which is again easy, interesting and important as well. So the chapter name is what, guys? Cell cycle and cell division. That's the name of the chapter. So before starting this chapter, I would like to ask few questions from all of you. And my first question is, how's your preparation going on? That's my first question. How's your preparation going on, everybody? Yes, everyone, how's your preparation going on? Going well, going great. If it is so, then it is a good thing. If it is not, then please start your preparation. You still have time left in which you guys can prepare. You can follow a proper strategy and you guys can really score very, very, very good marks. Okay, right? So cell cycle and cell division, this is our chapter name. So if I have to start with the cell cycle and cell division, I'll start with a very simple example. Let's take the example of a single cell. Okay, let's take the example of what? Let's take the example of a single cell. Students, if I take a single cell, let's take amoeba. You know that this amoeba, right, throughout its life cycle, this amoeba will grow, it will divide into two, isn't it? It will divide into two, isn't it? So this one will be the parent cell and this is going to be the daughter cells, right? Parent cell, and here we will have the daughter cells. So your cell is going to grow. This is a case of a unicellular organism which is growing and which is dividing into the two. So this is one case. I'll give you the cases here. Now take the example of the zygote. If you take the example of the zygote which is 2n, 2n means diploid. This zygote, you know that it will develop into a complete organism. Let's take complete human being, right? In a complete chintu, this single cell zygote is going to develop, right? We know that this zygote can develop into a complete multicellular organism. Here also there is the requirement of something which is known as division. This single cell needs to divide, then it has to increase its number, and ultimately it will result in the formation of a multicellular organism, isn't it? So even if I talk about starting the life from a single cell, that is zygote, or if I talk about a unicellular organism that is existing there in the nature, they all are, with time, they all are growing, with the time they all are dividing, isn't it? With the time they all are growing, with the time they all are dividing, isn't it? So somewhere we can clearly relate that division is something very, very, very important, isn't it? Division is something which is very, very, very important, isn't it? Right, isn't it? So now when I talk about the organism, right, indeed in the living organisms, be it a unicellular organism, be it a multicellular organism, growth and reproduction, they are most important, right? They are one of the most important features of living being, isn't it? They are one of the most important features of living beings, a very important quality, or I would say that this is the fundamental quality here, fundamental property here. Am I right? This is the fundamental quality here. This is the fundamental property here. Am I right? Yes. So that is what we see here, isn't it, guys? That is what we see here, isn't it, guys? So this, for this growth and reproduction, you definitely need this thing which is known as cell division, right? You need something which is known as division for this growth and reproduction, especially in the case of unicellular organism. Because look at this unicellular organism, the parent cell, the daughter cell, one cell is dividing into two. Yes, one cell is dividing into two. A single parent cell is giving me the two daughter cells. So in the case of a unicellular organism, right, when it is growing, it is showing the cytoplasmic growth and then it is dividing into two. So here, in the case of single-celled organisms, right, in the case of single-celled organism, I can clearly see that there is no difference in growth and reproduction. They are taking place simultaneously. So you guys can see how important this division is, because growth is taking place, then the division is taking place. That is what we are calling as reproduction here in this case. So division is something very, very, very important, isn't it, guys? Division is something very, very, very important. So do you people think the same? Yes. Do you people think the same? That yes, ma'am, division is something very, very, very important. Now you all need to answer one of my questions, and that question is, why do a cell divide? Okay, fine, I got it. Division is important. I got it. A single cell, that is zygote, is dividing. Normally, unicellular organisms, they are dividing. But my point is, why this division is important? Yes, people, why this division is important? Tell me. Tell me. Yes, please, tell me. Anybody? Why the division is important? So if I talk about that, why do a cell divide? Why do a cell divide? So we have the answer. Of course, we have the answer. The very first thing we need to answer here is surface to volume ratio. That's what we need to answer for, understand first. Right? What do we need to understand first? Surface to volume ratio. Second thing that we need to understand is nucleocytoplasmic, which is also known as kernplasmic ratio. That's what we need to understand, isn't it? That is something which we need to understand, isn't it? Yes or no? Okay, so let's start with this surface to volume ratio thing. Let's start with what, people? Let's start with this surface to volume. Right, let's start with what? Let's start with this surface to volume ratio thing. Let's talk about that. So, guys, you know we have the like whenever we talk about the cell, right? Do you know that we have small size cell as well? Yes, everybody. Do you know that we have small sized cells as well? Yes or no? Yes or no? See, when a cell is small in size, when a cell is small in size, its surface area is more in comparison to its volume. Literally. So imagine this is a single cell which is small in size, having dense cytoplasm, having conspicuous nucleus. We call these small size cells as active cells. We can also use the word immature cell for it. Now, when this cell will grow, students, right, when this cell will grow, obviously cytoplasmic growth is there. And what are we going to observe? That surface area, right, surface area is less in comparison to its volume. Are you getting my point? Surface area is less in comparison to its volume. Now here, because we have more surface area, right, in comparison to that, its volume is low. So of course, this more surface area will be sufficient enough to withdraw the material from the outside, right, for the exchange of material. And that is how the complete functioning here, all the needs of the cell will be fulfilled. Now, when the cell is going to increase its size, or when the cell has increased its size, its surface area in comparison to its volume is less. Due to which, right, this surface area, right, due to which, of course, the exchange of material will be hampered, and due to which that less surface area will not be able to, because exchange will not be at that rate. Okay, so because of that, what is going to happen, the needs of this particular cell will not be fulfilled, and that is also a sign for the division. Next is nucleo to cytoplasmic ratio. Next point is what, students? Yes, everybody, do write it down. Nucleo to cytoplasmic ratio. Now, when a cell is small in size, again the same story. When a cell is small in size, my dear students, what you people will see, it contains a prominent nucleus, and comparatively, we'll see less cytoplasm is there. But students, when cell grows, cytoplasmic increase is there. What is that, students? Cytoplasmic increase is there. So when cell grows, right, its cytoplasm increases. Its cytoplasm increases. So now that nucleus, okay, now look at this small size cell. Look at the nucleus size here. This is a large size cell. Look at the nucleus size here. So of course, now this nucleus is not sufficient enough to, control all the activities of this particular cell, because the cytoplasmic growth is more. So when, of course, so here what you need to keep in your mind, that basically when a cell will grow, there is a disturbance in the nucleocytoplasmic ratio. Because when it is small, less cytoplasm is there, nucleus can efficiently control, right here. So prominent nucleus, less cytoplasm is there, right? So, just a minute. So, nucleus can efficiently control the activity of cytoplasm. It can efficiently control the activity of cytoplasm. But when size increases, students, then cytoplasm is more and nucleus is inefficient. Nucleus is inefficient to control cytoplasm's activity. So that is how the ratio will be disturbed, and this disturbance in the ratio is a sign that the cell needs to divide. Is that clear? Is that clear, everybody? So that is why a cell divide. Okay, that is why a cell divide. Okay, so these are the causes of cell cycle and cell division that even I have mentioned. You guys can get it in our official Telegram group as well, right, students? So I hope this is clear to you that division is very important to restore these balanced ratio, and on the basis of that, right, of course, for that the nucleus is basically going to do, the cell is going to divide. Okay, now we have to start this chapter. So I will discuss few basic terms so that you guys will have an idea that what is going on. See, in our body, I'm not going to write the definitions here. I will just use few terms, and I hope you all are aware. Firstly, I will talk about the somatic cells. These are our normal body cells. Like for an example, if I take the example of the humans, right? In the case of humans, if I am talking about the cells that forms the gamete, students, the name of that cells are germ cells. Germ cells are gamete forming cells. Now, if you are not aware what gametes are, and I'm sure you all are aware, gamete forming cells. Gametes, basically, they are the sex cells. Basically, you know that here we are talking about the testes and we are talking about the ova. sorry, we are talking about the sperm and we are talking about the ova, my bad. Okay, this is what we are discussing. These are the gametes. These are the gametes. So when you talk about these gametes, so you know it very well that in the humans, there are germ cells. That germ cells will give us the gametes. These are the cells which are going to form the gametes, which are the haploid cells. So these gametes are what? They are haploid cells. Haploid cells. They just have one set of chromosomes. One set of chromosomes. So you know that when there is the fertilization, they both will fuse, you will get the zygote, and then further growth will be there. So zygote is basically diploid. So when, let's say, human is developing, these are our normal body cells. Normal body cells, right? They are having no role in the sexual reproduction. These are the somatic cells, like let's suppose cells of my hand, cells of my face, cells of my other cells of my body, other than the sex organs, other than the cells which will form the gametes. So they are the somatic cells, and another term is the germ cells. So germ cells, gamete forming cells, where do you find them? We find them in the gonads. So what are gonads? Gonads are nothing, students. They are the primary sex organs. So these are the 3 or the 4 terms that I have used. Secondly, yes, we will talk about the word generation time. Yes, we will talk about the word generation time. So anybody, yes, anybody who can tell me the definition of the generation time? Anybody who can tell me the definition of the generation time? Anybody? Anybody here who can tell me the definition of the generation time? So when I'm using the term generation time, so obviously it is a time in between the 2 successive generations. Successive means one after another. Or I can tell you it in this way. Let's suppose this is a cell. This cell is taking 20 minutes to divide into 2 daughter cells. Right? This cell is taking 20 minutes to divide into 2 daughter cells. So of course, these daughter cells will further divide, and they will also take another 20 minutes. So this 20 minute is the generation time here. What is this 20-minute generation time here? So can I not say that this is the time in between the 2 successive generations? Or can I not say that it is the time taken by a cell, right? It is the time taken by a cell to form its daughter cells, isn't it? Time taken by a cell to form its daughter cell. So can we say it like this? Yes, everybody. Can we say it like this? Yes. Yes, please. Can we say it like this? So I can also define it in this way, that it is the average time between 2 successive means one after another, 2 successive generations. So we are calling it as generation time. Like in the case of E. coli, it will take 20 minutes to form the progeny. So this is what the generation time is. So these are the terms that we will use: generation time, gonads, which are also known as sex organs, gamete, haploid cell. When I'm using the term haploid cell, so I am talking about the cells having 1 set of chromosomes, diploid cells having 2 sets of chromosomes. So these are the terms that we are going to use to understand this particular chapter. So anybody having any doubt here? Anybody having any doubt here? Now, this is a generation time. As I said, the time interval between successive cell division, or 2 successive generation. Now, beta, this type depends. It depends upon the type of cell. It depends upon the type of organism. Like for an example, if you people talk about the bacteria, the structure of bacteria is very simple. You know that bacteria, they just contain nucleoid, right? So in their case, this nucleoid, this genetic material will grow, cytoplasmic growth is there, and they will divide into 2. So the structure is simple. So time will also be less. But if you talk about a eukaryotic cell, it will take more time for the division in comparison to this bacterial cell. So this is the generation time. And as per the cell, as per the organism, this generation time, it varies. For an example, right, here you can see if it is bacteria, they'll divide faster in comparison to a eukaryotic cell. So temperature, oxygen availability, food, reproduction, everything affects this generation time. Right? Everything affects this generation time. So it is different. It is different for different organisms. Right? It is different for different organisms. Now, the next thing that we need to understand is basically what? It is the cell cycle, students. Right? Now, this is something that we need to understand. What? The cell cycle. What exactly is the cell cycle? This is the definition that you all can see. That it is the sequence of events. It is not one event. Right? Let me highlight this point. in general, when you are talking about the cell cycle, you're simply saying there is a cell, this cell will grow, because growth is that ultimate reality. Growth will be there. There is a cell, that cell is growing. Its cytoplasm is increasing. Are you getting my point? Its DNA will replicate, cytoplasm will increase, DNA will replicate, all that things will occur in a sequence. And after that, a cell will divide into two. Are you getting my point? After that, a cell will divide into two. That is the same story here. So what is the important thing? That this is not a single event. It is a sequence. It is a sequence of events, and all that events are taking place in a coordinated manner. It's not like that there is some random event that now the cell is increasing cytoplasm, now the cell is increasing the DNA replication. Sometimes it is doing DNA replication first or cytoplasmic growth is later. No, there is nothing like that. Are you getting my point? Are you getting my point? So it is a sequence of events by which a cell is going to duplicate its genome, right? A cell is going to duplicate its genome, means genetic constituent, right? Genetic constituents. It will synthesize. This definition is important, and again, you know that it's our NCERT. So it synthesizes the other constituents of the cell. Other constituents means we are basically talking about the other organelles, other enzymes, protein, everything we will consider, and then it will divide into two daughter cell. So this is what cell cycle is. So what is it, everybody? Pay attention here. What is it? It is a sequence of events by which a cell is going to duplicate its genome, it is going to synthesize its other constituents, and eventually it will divide into two. This is what the cell cycle is. Am I clear here? Am I clear? So during the cell cycle, or when you talk about the growth of the cell, one thing that you all are going to see, that cytoplasmic growth will always be there. Are you getting my point? That cytoplasmic growth will always be there. Are you getting my point? You can see this, कि although cell growth in terms of cytoplasmic increase is a continuous process. Like they are relating it with the other point, but you have to pay attention here that ultimately the cell growth will be there. Cell growth means cytoplasmic growth is there. Ultimately, it will be there. It will be there. But when it comes to the DNA synthesis, it is very specific, students, because it is going to take place only during one specific stage. So can I say that when it is the cell cycle, there is a sequence of events, everything in a coordinated manner, there is a continuous cytoplasmic increase, but DNA replication will be there in a one, or I would say particular stage, right? So these are replicated chromosomes, right? These replicated chromosomes or the DNA will then be given to the daughter cells, right? And then again by the complex series of events. So this is not something that, okay, fine, the chromosomes, they just duplicated their arms and then they will be distributed to the daughter cells. No, not like that. Again, there will be the series of events. So everything we will read under what? We will read under the cell cycle, and as I said, everything is under the genetic control. Are you getting my point? Everything is under the genetic control. Now, let me explain you one more thing. That is the genome. No doubt, I clearly mentioned it, that genome is basically the haploid content. What is genome, my dear student? Genome is the genetic constituent. This is what I just mentioned. Yes, please. This is what I just mentioned. That genome is nothing. Genome is the genetic constituent, genetic composition of that particular cell. Now, students, understand one thing. If you talk about a diploid cell, right? A diploid cell, that is 2N, which is having two sets of chromosome. One is from mother's side, another is from father's side. They are, of course, going to have the same genes. The allele might vary, but genes are going to be same. So they have that same genetic information. If I'll check the chromosome 1 from mother or chromosome 1 from father, at least it can tell me that which genes are placed on that chromosome and where are they placed, what is the sequence. So yeah, sometimes for the genome, we also say that it is the haploid content of the cell, haploid genetic content of the cell that we are studying, from where we are getting the genetic information. Is that clear? Is that clear, everybody? So anybody having any doubt here? Anybody having any doubt here? So that's something you have to keep in your mind. And here you people can again observe this thing, that it is the period of cell cycle which varies from organism to organism. For humans, it is 24 hours. For E. coli, that is a bacteria, 18 to 20 minutes. And yeast, which is a unicellular fungus, Saccharomyces, it is for this approximately 90 minutes. So it varies, as I said. Okay, beta? Okay? Yes? Right. So let's go to the next thing. This is the next thing that we have to focus, right? And here we are going to make it together. So when students, when we talk about the cell cycle, when we talk about what? When we talk about the cell cycle, the very first thing that we are going to read is interphase. Yes, we will talk about the interphase, then we will talk about the M phase. Interphase is there, M phase is there. Now, this interphase is also known as resting stage. This interphase is also known as I phase, invisible phase, energetic phase, anything that you guys can use here. So it is interphase, resting stage, I phase, invisible phase, energetic phase. Even it is also known as preparatory phase. It is also known as preparatory phase. And when you talk about this M phase, it is the actual division phase where the cell is going to divide into two. Is that clear? Right? So now in the interphase, yes, you will have G1 phase. That's what you have to study. You have to study the S phase here. You have to study the G2 phase here. Are you getting my point? So G1 is growth 1 phase, gap 1 phase, pre-DNA synthesis phase. You need to know the another names, guys. Then there will be the post-mitotic phase, right? Post-mitotic phase. This is what your G1 is. So G2, you can also call it as second growth phase or growth 2 phase it is. You can also call it as gap 2 phase. You can also call it as post-DNA synthesis phase, post-DNA synthesis phase, and you guys can also call it as pre-mitotic phase. That's the name that you people can use here. Now when it comes to the S phase, so it is the synthesis phase. Phase where DNA replication occurs. Exactly. NTA's favorite question. Okay, this is the NTA's favorite question. Always they are going to ask it. So there will be the DNA replication. What will be there? There will be the DNA replication. This is what your S phase is. Moreover, centriole duplication occurs here. So this is what you need to do. Firstly, we will read it, right? Firstly, we have to read it. We have to understand it, right? So this is what you have in your interphase. I'll explain each and every line written there. Now, M phase, actual division phase. M here is, of course, mitosis. So here you will see there will be karyokinesis, students. There will be karyokinesis, then there will be cytokinesis. Are you getting my point? So in the M phase, what you people are going to study? Yes, everybody. In the M phase, what you people are going to study? Karyokinesis. So karyon means nucleus here. Kinesis means division here. So there will be the division of nucleus, right? That is what we have to study here. Under this, we will study prophase, metaphase, anaphase, and telophase. When it is the cytokinesis, cytoplasm. So it is the nuclear division, students, and this one is the cytoplasmic division. Nuclear division, and here you have what? You have cytoplasmic division. Am I clear here? So this is what we have. This is what we have to read. Now there is a line, right, that we need to understand, that interphase. There is a point that we need to understand, that interphase is a phase between two successive M phases. So interphase is a phase between two successive M phase. So what's the meaning of that? This is the M phase. This is the M phase that we have. This is the M phase that we have. This is the I phase that we have, isn't it? This is the I phase that we have. So of course, M phase, and I phase. That's what we have. So I phase is having G1, I phase is having S, I phase is having G2, isn't it? So see this, G1 is coming after mitosis. That's why we are calling it as post-mitotic phase. Can you see that? So basically we are decoding each and every single line, right? Each and every single line of NCERT. So please tell me, are you able to understand it, guys? Are you able to understand it? Everybody? Yes? So post-mitotic, so of course, G2 will be pre-mitotic phase. Pre-mitotic phase. Or can I not say that G1 is actually a phase in between division and DNA replication, because I told you DNA replication will be there in only one substage, and that is your S phase. Is that clear? Is that clear, everybody? Please have a look. Please have a look. Done? So now let's talk about the interphase. Let's talk about the interphase. So as I said, yes, as I said that interphase is a phase which is resting. But I am also saying it is the energetic phase, but I am also saying that it is the preparatory phase. So what exactly it means? See, when our cell needs to divide, it needs to prepare itself for the division. Like you people are preparing for the NEET examination. You guys are giving your 2 years, 3 years, sometimes 4 and sometimes 5 as well, okay, for cracking this particular exam, which is just for 3 hours, isn't it? Which is just for 3 hours. You guys are putting your effort so that you can just crack it, isn't it? So that you can just crack it, isn't it? Isn't it? So now when you are talking about this particular interphase, okay, so here in this particular phase, a cell needs to prepare itself so that it can just divide without any error. Are you getting my point? Like you are preparing, you are giving your best, you are putting all the efforts. For other people, you are doing nothing. You are just in 11th or in 12th, and then you took a drop year, you're just studying, you're doing nothing. For other people, it is like that. But you people know, you guys are under immense pressure. Right? You people know that that preparation time is so disturbing it can be. No doubt we enjoy it, but it is disturbing as well, because we have to be very focused. We have to make sure that we put all our efforts so that we can crack that exam. That is just for 3 hours. Just imagine. So for that 3 hours exam, right, we spend our 2 years, 3 years, 4 years, sometimes 5 years, so that we can do wonders in that particular exam. Same is the case here. Your cell needs to divide. It needs to do it without error. It needs to put because if there will be error, it can result in mutation. So in the interphase, it is preparing itself for that particular M phase, which is demanding a lot of energy. The accuracy should be there, right? So interphase is the phase in which it is preparing. But when you people observe the nucleus of the interphase under the microscope, you are not going to see some, movement of chromosome or something like that. You people will see that not even the chromosomes are there. Chromatin is there. It is also tangled, right? It doesn't show you some activity. So sometimes scientists were like, this is a resting stage where nothing is happening. But later on, right, we got to know that it is the phase where cell is actually metabolically very active. Are you getting my point? So this is what interphase is. This is what interphase is. Or remember, a cell spent more than 95% of its lifespan in interphase. Imagine. This is what interphase is going to do. This is what interphase is going to do, right? This is the duration of the interphase. A cell will spend more than 95% of its lifespan in the interphase. Like for an example, in the case of humans, I'll give you the example. So I will give you the example of the humans. So in humans, as I said, the division will be there within 24 hours. A cell will divide and it will take 20. That total duration is basically the 24 hours. So in that 24 hours, right? In that 24 hours, the cell division, that M phase cell division is for less than 1 hour. I won't say even more than 1 hour, for less than 1 hour. So imagine, rest is just the interface. Rest is just the interface. So any doubts here? Any doubts here? So now we have to understand that what exactly is the G1 phase, what exactly, right? We need to understand that what exactly is the G1 phase. See this. Interphase is present in between two successive M phases. So M phase, interphase, M phase. So that is what we need to discuss. So here you people can see, as I said, guys. Okay, let me arrange my pen. So it's a long non-dividing or growing phase of the cell. It's a long non-dividing growing phase of the cell, isn't it? It's a long non-dividing growing phase of the cell. So it includes cell growth, it includes DNA replication, and it is the most active stage because there will be intense biosynthesis will occur here. So it is a phase where cell is growing. It is a phase where DNA is replicating. So it is the phase where the cell is metabolically very, very, very active. Isn't it? Isn't it? So this is what we have to understand first, that what exactly is the G1 phase, right, students? What exactly is the G1 phase? See, as I said, it's a phase, right? It's a phase between what? Between cell division and DNA replication. It's a phase in between cell division and DNA replication, number one. Secondly, G1 phase is most variable phase in terms of duration. Yes, you heard me right, students. When it comes to the duration of the, duration of this G1 phase, it varies. Do you know that? It is not fixed. You cannot say that it is for 1 hour, 2 hour. It depends. Like it depends. It literally depends. It can be for 1 hour. It can be for 2 hours. It can be more than that. It depends which cell is dividing, what is the requirement there. So it is the most variable phase. I'll give you one example. You all know that we all arise from a single cell, that is zygote, right? And because zygote is capable of developing into a complete organism, we also use the word totipotency. What is the word that we use there? We also use the word totipotency. So what exactly is the totipotency? It is the ability. It is the ability of a single cell to develop into a complete organism. This is what zygote is. When zygote divides, initially the name of the division that we use is cleavage. What is the word that we use for it? Cleavage. It is just like your mitosis only. But here in the cleavage, students, there will be no G1 and G2 phases. I'll tell you why am I telling you this. There will be no G1 and G2 phases. Now look at it. You are talking about interphase, where you are starting with a G1 phase. You are also talking about a division here, the cleavage which you are calling, and you are saying, right, and you are saying that there will be no G1 and G2 phase here. So see, here, not required, not there. Are you getting my point? That is also the case. So G1, and again, once again, I'll come to that. So you listen to this. So G1 is the most variable phase in terms of duration, number one. So what is going to happen here? Obviously, your enzymes, right? Enzymes, comma, proteins, RNAs, nucleosides, etc., ATP, so etc., they all will synthesize in this phase, right? They all will synthesize in this phase, right? Then, basically in this phase, can I say that cytoplasmic growth is there? Can I say that cytoplasmic growth is there? Yes or no? Yes or no? Yes? Yes, we can say that. Right, students? We can say that. Why? Because this is actually happening here. Right? This is the thing that is happening here. Are you getting my point? So any doubt? Any doubt? So cytoplasmic growth is there. Your enzymes, your proteins, your RNAs, nucleotide, whatever is required for the next substage. Everything, my dear students, everything will be synthesized here. Right? Everything will be synthesized here. Are you getting my point? Are you getting my point? Yes? Yes? So remember, here in this phase, your cell is metabolically very active. Metabolically very active. So That's why I already mentioned cytoplasmic growth is there, but no DNA replication. Remember this. But no DNA replication is there. Right? But no DNA replication is there. Are you getting my point? So DNA will only replicate in the S phase. NTA's favorite question. Keep it in your mind. Okay, that is your NTA's favorite question, and that's something you have to keep in your mind. Is that clear? Is that clear, everybody? Okay. So next thing is in G1 phase, your chromosomes are of course they are present. That chromosomes are in monad form. And now what is this monad form? Just allow me 1 minute, right? I'll explain it in the S phase. So they are in the monad form. Basically, they are having single arm. I'll come to that topic later. So now in G1 phase, cell is having two options, right? When a cell is in G1 phase, either it can enter in S phase, students, right? Or it can leave cell cycle and can enter in G0 phase, which is the cell arrest phase, which is the quiescent stage. That is the case here. Are you getting my point? That is the case here. So in G1 phase, either your cell, it can enter in S phase, or it can leave cell cycle and can enter in G0 phase. Okay, it can enter in G0 phase. Now I'll relate it with this cleavage wala part. So yes, I'll relate it with cleavage wala part as well, and I'll explain this point as well. Firstly, when you talk about the cell cycle, G1 phase, then comes the S. But it is not mandatory that all the cells will keep on dividing only, right? That is not going to happen. That's how the cell cycle is. You talk about the G1 phase, you talk about the S phase, you talk about the G2 phase, and finally, the cell will enter in the M phase. Now, students, when you look at this G1 phase, if there is no go-ahead signal, right? If there is no go-ahead signal for the division, the cell will leave this and will enter in one G0 phase, which is also known as cell arrest phase, which is also known as quiescent phase. Are you getting my point? If the go-ahead signal is there, it will enter in the S phase, right? It will enter in the S phase. Now, the another point. I was giving you the example of zygote. I was giving you the example of cleavage. I told you in cleavage, you are not going to talk about no G1, G2 phase. So G1, G2 phase, both are concerned with the cytoplasmic growth. So that is why in the zygote, initially when your zygote is dividing, DNA is replicating indeed, but there is no division of no growth of the cytoplasm. So that is why initially your zygote will only be there in the same size, because cytoplasmic growth is not there, size will not increase, cell number will increase because division is there. So M phase, S phase, M phase, S phase, M phase, S phase. That's what you can imagine keep going on. But there will be no cytoplasmic growth. That's it. Now, let's move to the next point, S phase. Right now, I'll explain it. So kindly listen to me very carefully. S phase, right? Your synthesis phase. The phase in between G1 and G2 as well. So it's a phase in which DNA replication or duplication, whatever you want to call it, DNA replication or duplication occurs. That is, if initial amount of DNA is 2C, then it will become, my dear students, then it will become 4C. So can I not say that initially DNA is 2C here, then it will become 4C. But is it the important thing? Yeah, it is important because DNA replication is taking place here. But the another important thing is, but number of chromosomes will remain same. So try to understand this part. Chromosome number will not be altered, will not be doubled, it will be same, but it is just the DNA amount that will get doubled. This is the very important point, right? This is the very important point, right? Next point that we have here is, this is the 1 point, students. Next point that we have here is, right, that you need to remember that centriole, which is a non-membrane bound organelle, which is present in animal cell. We have discussed it. So centriole duplication also occurs here, right? Even the centriole duplication also occurs here. Is it clear? Yes, everybody. So I hope you guys are making the notes as well. So is it done? Is it done? Moreover, because DNA duplication is here, so histones and non-histone protein synthesis, histones and non-histone proteins synthesis occurs in this phase. Yes, even your histone and non-histone proteins will also get synthesized here. Am I clear? Am I clear? So I am saying, right, I am saying that firstly there is a DNA replication, that if the initial amount of DNA is 2C, it will become 4C, but the chromosome number will remain same. I am saying the synthesis of histone and non-histone proteins will be there. In case of animal cell, centriole duplication will occur here. But this point I need to clarify, that why is it so? Why DNA is replicating, or duplicating, but still there is no chromosome, there is no increase in chromosome number. This is the point which we have to understand. Just a minute. Right, this is the point that we have to understand here. Okay, so I'll give you an idea. See, no doubt in the interface, no doubt in the interface, when you look at the genetic material here in the interface, indeed all that genetic material, right, all that genetic material, indeed that is basically tangled, right? All that chromatin is tangled. You cannot say that it is discrete, but because I have to explain you this topic, so I am asking you people to assume one thing, right? I'm asking you people to assume one thing. Assume a cell having 2n is equals to 4. Just giving you an assumption. Assume a cell is having 2n is equals to 4. Just imagine that this particular cell, obviously, basically, I'm trying to tell you that this cell is having how many chromosomes? This cell is having 4 chromosomes. So this is going to be the case here, students. This is going to be the case here. This is going to be the case here. Keep it in your mind. Right? 1, 2, 3, 4. So in the G1 phase, I told you clearly that the chromosome is in the monad form. This is what I'm trying to say. Basically, when we hear the word chromosome, right, when we hear the word chromosome, so we just think of this structure only. This structure only. We only think of this structure which is having, you know, 2 sister chromatids attached to 1 centromere. We think this is the chromosome and we think it is the chromatid, but actually that is not the case. This is also your chromosome now. This is also your chromosome now. This is basically, it is having single arm. It is having single arm. That's why you are using the word monad here. That is why you are using the word monad here, isn't it? Isn't it? That is why you people are using the word monad here, because there is a single arm only. You don't have 2 arms here. You don't have sister chromatids here. And ultimately, if you people talk about the chromosome, what exactly is this chromosome? Right? It is your chromatin. What exactly is chromatin? It is just the DNA. DNA coiled over some proteins. DNA coiled over some histone proteins and then some non-histone proteins. Actually, they all together are making this structure. So if I'm talking about this structure, I'm talking about nothing but the DNA and proteins only. It is just the DNA and proteins. Here you have the centromere, primary construction in between. Are you getting my point? Are you getting my point? So you have the DNA and protein only which is making it, but here there is a single arm, so you are calling it as monad, right? Now in the S phase, because DNA duplication is there, right? DNA duplication is there. This is the meaning of DNA duplication. So ultimately this chromosome itself is DNA. It is itself that protein, so it will get duplicated. That's why some students get confused. Ki, ma'am, chromosome number is same. Then why do we say that chromosome is getting duplicated? So actually it is made up of DNA proteins only. Right? Right? it is actually DNA and protein only. So because it is actually DNA and protein only, so this is getting duplicated, this is getting replicated. So in S phase, you will see that your chromosome is having two arms, right? They are basically the sister chromatids attached to a centromere. The centromere is holding these sister chromatids together. So now, if I took the so here I took the example of a cell which is having 4 chromosomes. So see, that is how I'm showing 4 chromosomes, 1, 2, 3, and 4. So after the division also, so in the S phase also, the chromosome number is still same, right? The chromosome number is still same, students. It is what? It is still same. Look at this, 1, 2, 3, and 4. Here also, chromosome number is 1, 2, 3, and 4. But look at the arms, they are duplicated. Look at the chromosome, it is duplicated. So here, the DNA amount is 2C. Here, the DNA amount is 4C. Here, your chromosome is monad. Here, your chromosome, it is having 2 arms. So you will use the word died. You will use the word died. So that is why we say that the chromosome number is same, but the chromosome number is same. It's just the amount of DNA which will get doubled. Am I clear? Yes. So that is how we are saying that chromosome duplication is occurring. Right? So when we are using the term chromosome duplication is occurring, we are saying it is the DNA content of that chromosome which is getting doubled. Am I clear? It is the DNA content of that chromosome which is getting doubled. Any doubt, guys? Any doubt, guys? Sure? Sure? So can I not say that it is the S phase in which your monad chromosome, monad changes to diad? Now, I hope you all are able to understand it. That it is the monad that will changes to diad. It is the monad that will changes to diad. So, next is G2 phase. Again, RNA, protein, etc. synthesis occurs here. Right, students? Here you will see the synthesis of tubulin protein. Right? Synthesis of tubulin protein. So this is the protein that will form microtubules. That microtubules are important for the spindle fiber formation. Is that clear, students? That is important for the spindle fiber formation. So cell size, nucleus. So ultimately here also cytoplasmic growth is there. Ultimately here also cytoplasmic growth occurs. Right, students? So RNA, protein, etc. Their synthesis will occur. Synthesis of tubulin protein is there. So cell will, right? In the cell you will see, in the cell what will you see? The energy store increases, right? So ultimately cell size, nucleus, cell size, nucleus size, and energy store increases. Are you getting my point? Energy store increases. That is what your G2 phase is. Are you getting my point? That is what your G2 phase is. Okay? Understood? Understood. So look at this particular diagram. Just have a look at it, right? You have G1 phase. Look at this. Look at the duration part here. Now, here you have a point, students. We call that point as restriction point. Like in some organisms, we call it as restriction point. In some organism, the same is known as start point as well. Right? In some organism, it is the restriction point. In some organisms, it is also known as the start point, my dear students. So basically, what is going to happen? Right? Basically, what is going to happen? So when there is the G1 phase, so if cell will get a go-ahead signal that yes, I have to cross this, I have to go for the division. So if cell has crossed this restriction point, then definitely it will enter in the S phase. But let's say if there is no go-ahead signal, but let's say if there is no go-ahead signal. So yes, you can see this. If there is no go-ahead signal, the cell will exit this phase and it will enter in a phase which is known as G0. Which is known as G0. So G0 is also known as the quiescent stage. It is also known as the cell arrest stage. It is also known as the stage in which, so cell is, though we also call it as, sometimes we call it as inactive stage, but inactive stage in terms of what? In terms of proliferation. So G0 phase is a phase in which cell is metabolically active, in which cell is metabolically active, but it's not proliferating. But it's not proliferating. Are you getting my point? But it's not proliferating. That's what we have to keep in our mind. Okay? So this G0 phase, it can be temporary, like for some time cell is not dividing, so cell will enter in this phase. Then it can be permanent as well. Then it can be permanent as well, right? Like in the case of your heart cells, in the case of your nerve cells, they divide. When do they divide, students? Heart cells, nerve cells, when do they divide? They divide, right? Only when, right? These heart cells, permanent cells, heart cells, nerve cells, yeah, they do divide, right? They do divide, but when at the time of initial growth only, at the time of that embryonic stages only. Later on they don't. So they will be in the permanent G0 phase. And when it comes to the temporary phase, matlab they are there for the some time. Like right now the division is not required. For an example, let's suppose if you have an injury here, obviously cell division is required for the healing, right? Cell division is required for the healing. Then your cells will divide. Otherwise, why should they? Are you getting my point? So that's something you have to keep in your mind. Now, we will discuss one more topic, students, which is known as checkpoints, or we can also call it as, with the help of this diagram only, we will discuss it. It is the regulation of cell cycle. What do you call it, people? You will call it as regulation of cell cycle. This is what we have to discuss. So, regulation of cell cycle will include what? It will include some checkpoints. The topic of checkpoints is not given in NCERT, but yeah, it is important. So what are these checkpoints? See, remember, our body is a wonderful machine. In our body, we have the different systems. We have the different systems which will keep a check, right, on other systems as well, whether they are functioning well or not. Like let's suppose if in our body high blood pressure is there, so obviously we have, right, we have the hormones, we have a proper control that will, you know, stop it. If there is low GFR, then we have the system which will increase it. So there is a counter for everything if something is not working well, isn't it? Isn't it? Now, what is my point here? I am talking about the cell checkpoint. I am saying that let's suppose if your cell is dividing, right, we have to make sure that it divides properly. We have to make sure that errors should not be there. Why? Because errors means mutations. Some slow mutations are there in our system, but we cannot, you know, in our systems, the frequent mutations should not be there. So we have the checkpoints there. Firstly, so I'm talking about this G1 to 6 S checkpoint, right? Firstly, what am I talking? I am talking about this G1 to S checkpoint. Which checkpoint am I talking? The main checkpoint, G1 to S. Because this checkpoint will make sure that cell will divide. So how this checkpoint? So see, here what will be checked? Are we ready to divide? Matlab, is the cell is big enough? Is the cell is having sufficient energy? Is the cell is having other resources? Is DNA is damaged or not? If everything is well, right? If there is no problem at all, then there will be a transition to the S phase. There will be a transition to the S phase. Are you getting my point? So G1 to S transition. So students, growth factors, nutrient mediums, right? Growth factors, nutrients, energy stores, right? Energy stores, they all, etc., DNA damaged or not, they all will decide this transition. Is that clear? They all will decide this transition. Is that clear? Is that clear? And for these transitions, you will see, we discuss the role of some cyclins, which are proteins, right, and some CDKs. So CDKs are what? These are cyclin dependent kinases. These are cyclin dependent kinases that we have here. So they are periodic, you know, making, breaking is there, different cyclins are there, cyclin A, B, C, D, all of that cyclin dependent kinases, which are the enzymes. So periodically their breaking is there, that is going to control this. This is what we discuss here. So main transition, I told you already, this is going to be your G1 to S. Is that clear? That is going to be your G1 to S. So next is G2 to M transition. Right, G2 to M transition will also be there, students. Right, so here you will see all the chromosomes, they get replicated correctly or not, is the DNA is damaged or not, is the cell size is proper or not, that's what we have to check. Then in the M phase, right, here also beta we have the checkpoints, right, like here, right, basically metaphase to anaphase, we have one anaphase promoting complex as well. So there it will be checked that all the chromosomes, they are arranged in the center properly, are they ready to get separated or not. So when we move from metaphase to anaphase, which comes under the karyokinases, right, there also we have the checkpoint. So yes, we do need to remember this, right, it is something that should be very clear in our mind. Okay, so now let's draw some conclusions. So G1 phase, DNA amount 2C. Let's suppose if 2n is equals to 4, here also it will be 4 only. S phase, the DNA amount will be 4C. But what about the chromosome number? It will remain same. What about G2? G2 will also have DNA as 4C and the chromosome number will be 4. So yes, that's something that should be clear to you. That's something that you people should know. Yes? Understood? Sure? Sure? Are you sure, everybody? So if there is any doubt, then do let me know. Okay? So let's answer this. How do plants continue to grow? How do plants? You know that plants, they will keep on dividing. They have the meristematic tissue. But in the case of animals, we do not have meristematic tissue. So growth is only up to a certain age. So now, can you tell the name and location of tissue having cells that divide all their life? In higher plants, meristematic tissue, they'll keep on dividing, right? So animals do not have the meristematic tissue. So this is the NCERT, which should be very clear to all of you, right? So now you guys can see this part also, that you have studied mitosis in the onion root cell. Remember this, chromosome number in the onion. It has 16 chromosomes in each cell. So 2n is equals to 16, isn't it? So can you tell how many chromosomes will the cell have at G1? 16. After S phase, 16. After M phase, 16. You're talking about the mitosis only. Daughter cells will also have same chromosome number just like parent cell. But if I talk about 2C, so G1 will be 2C, S phase will be 4C, and even G2 will also be 4C. Is that clear? Is that clear? So that is something which should be clear. So see this, DNA content of the cell at G1, after S at G2, and after S will be 2C, 4C, 4C respectively. If the content after MC, it will become, after MC, it will become 2C. Because both the daughter cells will have same number of chromosome and this DNA content will be 2C, 2C. Right? We will discuss it in the mitosis. When we will start the mitosis, then it will be clear to you. So here you guys can see, right, these different types of cell checkpoints as well. So you people can see it here as well. G2 to M transition is triggered by MPF, maturation promoting factor, right? Maturation promoting factor, which will trigger the transition of G2 to M. That's it. So I hope this point is clear, right? I told you G1 to S is the major checkpoint. That is also an important question. G2 to M is the minor checkpoint. That is also an important question. So keep it in your mind, guys, right? So keep it in your mind. So this is all about, right? This is all about the cell cycle, right? This is all about the interface, we have discussed that in detail, right? You can just simply follow this content. This is going to be very, very, very useful for your NEET examination. There is nothing beyond this that you have to do. But yeah, one thing that you definitely need to do is MCQ practice. So keep practicing it. You guys can just join our batch as well, right? So here you people can see there will be the link for our batch. Okay, so you can click on it. You can be the part of our batch. Even on Unacademy platform, we take the special classes, right, that are free of cost, that are literally free of cost, my dear student. There you can practice the question. You can even study many lectures. You can simply subscribe to my profile there. You will get all the lectures. You can go for the revision. So I hope you liked it, right? I hope you like it, right? I hope you enjoy it, right? So see you all then.